We get confirmation that it started. Ding. All right, we are rolling. So let's get back to protists. So that's all I'm going to say for now about ciliates. Let's move on to dinoflagellates, which look rather different. Instead of multiple cilia, they've got typically just two long flagella. By the way, there's no real difference in structure between cilia and flagella, except that cilia are short, and usually you've got lots of them, and flagella are long, and usually, but not always, you have a smaller number of those. Cilium means eyebrows, by the way, so cilia look like, eyebrow hairs. And flagellum is Latin for whip. So think eyebrows versus whips. But there's no difference in structure between them. Lynn Margulis used to argue that being the case, they should both be called by the same word, and she proposed the term undulapodium to mean eukaryotic cilium or flagellum. But again, this is somehow never really caught on, except for Margulis herself and people that have followed her. So if you encounter the term undulapodium, now you know what it means. It's a eukaryotic cilium or a eukaryotic flagellum, not bacterial flagella, which are very different in structure. Anyway, dinoflagellates have a pair of flagella, one of which is kind of wrapped around the cell like a belt. It's called a transverse flagellum, and the other one trails behind. The beating of that transverse flagellum causes the cell to spin as it swims, and that's what the name means. Dino is very close to the Greek root meaning terror, which is what dinosaur means, terrifying lizard. But in this case, it's not quite the same word. It's a homonymous word that means to twirl. So dinoflagellates are the whirling flagellates because of the spinning motion that they make as they swim because of beating of that transverse flagellum. Most dinoflagellates are capable of feeding. About half of all species are photosynthetic. But this is weird. Dinoflagellates did not evolve with chloroplasts. What they seem to have done is different lineages of dinoflagellates, or the ancestors of different lineages, have engulfed different species of other algae, sometimes green algae, sometimes red algae, but kept the chloroplasts. Kind of like, I don't know, this gets into really, gross horror movie territory where like you dismember somebody, but implant, your victim's organs in your own body or something like that. That's really yucky. But it's what dinoflagellates have done, and it's what some species are still capable of doing, where they'll phagocytose a species of alga and store the chloroplast and keep them. And that's actually happened several different times independently in dinoflagellate evolution. If you're curious, it's called kleptoplasty. Plasty is in chloroplast or plastid, and klepto meaning to steal, as in kleptomaniac. And so, yeah, dinoflagellates have actually used stolen chloroplast from several different ancestral sources. Most, but not all, they all have alveolar membrane sacs. In most, those membrane sacs are filled with cellulose, and that means that just under the plasma membrane, there's this kind of armor-like structure called a theca. And you'll see what that theca looks like in a minute. There are some dinoflagellates that don't have this. They're naked dinoflagellates. They don't have a theca. There are a few genera that are mutualistic symbionts in other invertebrates, or even sometimes in other protists. Dinoflagellates in the genus Symbiodinium live inside the tissues of several different types of invertebrate, but the most famous is probably reef-building corals. Tropical reef-building corals need very strong light and very clear water because they're dependent for food on Symbiodinium dinoflagellates living in their tissues, and I'll show you that in a bit. And then finally, population explosions of dinoflagellates cause what are called red tides. These can be very dangerous because some species, when they swarm, release large amounts of toxins. The water can become toxic itself, or if you feed on filter feeding organisms from the water, like oysters and clams in particular, they may have eaten so many dinoflagellates that they build up high levels of dinoflagellate toxin, and you can end up with diseases like paralytic shellfish poisoning, which is exactly what it sounds like, slow paralysis and numbness of the muscles after eating contaminated shellfish. Anyway, there's a picture of the theca of a dinoflagellate. It happens to be Protoparadinium latistriatum. Don't memorize genus names, by the way. That theca is made up of cellulose plates that are butted together at the edges, kind of like a mosaic. Each one of those plates was made in an alveolus, in one of these membrane sacs. And you can see again how you have these very thin sacs that fit together at the edges and butt together very tightly. Running around the equator of the cell, there's that depressed zone called the girdle. And if this was alive, one flagellum would fit into the girdle and wrap all the way around the cell, and the other one would trail out behind the cell. Down at the bottom of the slide is where the other flagellum would have come out. That happens to be a picture of another one that's fairly common in fresh waters. The central body is kind of triangle shaped, but for some reason, this particular species has long horns. Those are just extensions of the cell and, extensions of the theca. And then running around the middle, you can see that girdle. You can't clearly see the flagella in this image, but it's the beating of the transverse flagellum that makes dinoflagellates spin as they swim. It's dinos, not danos. There's naked dinoflagellates that don't secrete cellulose, and this is one of them. This is a carnivorous dinoflagellate called Noctiluca, Latin for shining at night, because this one is bioluminescent. This one's also really big. It gets up to 1 millimeter across, although most of the cell is vacuole. On one of them, down at the right and a little below center, one of them is sticking this thing out. That's not actually a flagellum. That's a tentacle. These have these extensions, tentacle-like extensions of the cell that they can use to feed. And then when red tides bloom, they can not only decrease the amount of oxygen in the water, but they can also produce toxins that are directly poisonous. So there you have some dead fish floating in water caused by a bloom of a dinoflagellate called Carenia brevis. And again, if you eat fish or shellfish that have fed on toxic dinoflagellates, you can come down with paralytic shellfish poisoning because some of these are neurotoxins. Another related disease is called ciguatera. This is one of several reasons why you're not supposed to eat oysters if the month does not contain the letter R. All right, don't eat oysters in May, June, July, or August. The reason being partly because in summer the oysters are spawning and they're not as tasty. The other reason is that's the likeliest time for red tides to make your oysters toxic. So there, I don't know if you knew that rule, but you're only supposed to eat oysters in months that have an R in their names. Of course, that's also when you're at greatest risk for being attacked by pirates, if the month has an R in its name. Sorry about that. Okay. There's one dinoflagellate that's been implicated in killing fish directly. It's affectionately known as the cell from hell. And blooms of this dinoflagellate, Pfiesteria piscicida, have not only been blamed for toxins that kill fish, but supposedly some researchers that got exposed to them have ended up suffering long-term neurological damage, memory deficits, and things like that. This is not something you really want to deal with if you can avoid it. But it now seems those are some lesions that were blamed on Pfiesteria. But it now seems that it actually is an opportunist, that what it does is feed on decaying fish tissue on ulcers that have been made by a different parasite, a water mold called Aphanomyces. Water molds, by the way, are often the Stramina piles, if you remember them. So it doesn't look like Pfiesteria is capable of, taking down and eating fish, but it's still not something you want to mess with. On a lighter note, there's actually several places in the world that have this, but the one that's probably easiest to visit, or will be once the damdemic slows down, is a bay on the island of Vieques, which is just off Puerto Rico. Now, for a long time, you could not visit Vieques because it was an Air Force bombing and gunnery range. And it's still dangerous to go on the island because there's a lot of unexploded bombs and things, a lot of UXO. But you can now take kayaking tours to Vieques. And Vieques, that particular bay I mentioned, just happens to have conditions that are ideal for supporting a permanent high population of Pyridinium bahamense, which looks like this. There you can see again those cellulose plates making up the theca. And when you get lots of Pyridinium bahamense together, they do this. Pyridinium bahamense lights up when you disturb it. If you make any disturbance in the water, they'll all simultaneously start generating light. And bioluminescent bay on Vieques has such a consistent population of these that it's become a tourist attraction. You can go out in the middle of the night or the late evening in kayaks, and, well, you can see there's that glowing bioluminescent trail behind your kayak, and you can see more luminescence where, the paddle is hitting the water. You can actually see this in many parts of the ocean, but bioluminescent bay is very consistent for it. I think in other places you don't necessarily get it every night, but there's at least one bay in Hawaii and probably more in other parts of the world that are consistently bioluminescent because of dinoflagellate populations like Pyridinium bahamense. We've got at least one student who is in the Bahamas. Yeah, drop me a line or, text or chat me something and let me know if you've ever seen this. I'm curious how common this is elsewhere. One more that's of commercial importance. Now, dinoflagellates are important in marine ecosystems. The ones that aren't toxic are, of course, food for everybody else. There's one in particular that directly impacts the human economy. I mean, other than the fact that they can make your shellfish deadly. This is called Perkinsus marinus, and it's closest to the dinoflagellates, but not quite within them. So it's sometimes put off in its own tiny group called the Perkinsozoa, and it's a major parasite of oysters. Oysters will, as they filter feed, they'll ingest a stage that has two flagella, like a dinoflagellate, called a zoospore. And you have on the left lots of zoospores, and you can just barely see their little flagella. On the right, that's a cross section through one. You can see one of the flagella as that elongated thing, and then the other one's been cut perpendicular to the axis. So you just see that circle. That's a slice directly through a flagellum. So anyway, the infective stage is this little biflagellated thing that looks a little like a dinoflagellate. And then Perkinsus will start dividing and multiplying inside the oyster tissues, causing a disease that oyster growers call dermo. So called because Perkinsus used to be placed in a different genus, Dermocystidium. And dermo causes the oyster's tissues to waste away. It decreases their ability to reproduce, and it certainly renders them unsellable because you end up with these pus pockets in the oyster tissues, meaning that even if your oysters can live with a Perkinsus infection, you certainly would not want to put them in restaurants. So there on the left, you've got some nice healthy oysters. That's what they're supposed to look like. At the right, you've got some oysters that have been infected with dermo. I don't know if you're personally big fans of oysters, but they are a, pretty valuable seafood crop. It's a, billion-dollar industry. Places like the Gulf Coast of South Louisiana or Apalachicola Bay in Florida or Chesapeake Bay in Maryland. A lot of the local economy may depend on the oyster harvest, and oysters can actually be cultured and farmed in many areas, and this can be pretty serious if you're an oyster farmer. So that's one that has pretty good sized direct economic impact on some areas. Right. Last group of the alveolates is apicomplexa. And what makes these apicomplexans is they have an apical complex. What the hell's an apical complex? It looks kind of like a nose cone. They're a set of organelles that attach to the cells of a host because all apicomplexans are parasitic. There are no free-living apicomplexans anywhere. And the apical complex will attach to the cell of a host, and in many cases, although not all, the apical complex can be used to penetrate into the host cell. Many apicomplexans, like malaria is the best known, actually spend most of their life cycle inside the cell of a host organism. Most apicomplexans actually contain something that looks like a chloroplast but isn't. It's called the apicoplast. Last I looked, we're not too sure as to what the apicoplast does, but it is necessary for life, and there are drugs that are being developed against apicomplexan disease that target the apicoplast because the cell can't live without it. Even though the apicomplast is not photosynthetic, because as parasites, apicomplexans spend their entire lives in darkness, right? They're never in a place where they get direct sunlight anyway. But still, it's present. and it seems to be evidence for common ancestry with dinoflagellates. And there's no cilia, no flagella. Some stages of the life cycle may be capable of crawling or may be capable of what's called gliding. They just kind of very slowly scoot over a surface, and it's not immediately clear how. But they don't swim. They don't have cilia, nor do they have flagella. They're all parasitic in animals. The big human disease caused by apicomplexans is malaria. There's actually 5 different species that cause human malaria. Toxoplasmosis is another one you might have heard of. Cryptosporidiosis, not, I think, normally that serious, but it can be really bad if you're immunocompromised. So babies, HIV patients, transplant recipients are at greater risk of cryptosporidiosis, which is you don't want to deal with because you can get it through drinking water if it's not properly treated. Coccidiosis in poultry is a economically important disease. Dogs and cats can also get coccidiosis. Something called babesiosis is a disease of cattle that fortunately is not present in the United States, but could always come back. We had to eradicate it. There's others that you'll encounter if you go on to medicine or veterinary medicine. And unlike parasitology, I'm not going to drag you folks through the entirety of the life cycle. Let's just say that for most of the life cycle, the parasitic state, the stage is haploid. Most of the life cycle is spent haploid. But at some point, 2 haploid stages will pair up. They'll form gametocysts, which is just round, hollow things with a pair of them inside. Sexual reproduction takes place within that gametocyst. So briefly, there's a diploid stage, but usually immediately after that, that diploid stage undergoes meiosis and produces new haploid stages again. So these have alternation of generations, superficially like plants. But the diploid stage is very short-lived, and most of the actual life is spent haploid. And many species will alternate sexual reproduction with asexual division called merigony. I think I usually say merogony. I don't know which is right. But, like Miley Cyrus, that gives you the best of both worlds, because I mentioned that sex is a way of generating diversity and genetic diversity and cleaning up DNA damage. So you get that. The advantage of asexual reproduction is it's faster. So many apicomplexans can increase their numbers very rapidly in a parasitized host and then switch over to a sexual stage to get the benefits of sexual reproduction. You get the best of both worlds. Chill it out, take it slow, then you rock out the show. Sorry, don't know what came over me there. Right. So there's a diagram of what the apical complex looks like in an apicomplexan cell. Don't worry about all this detail, but there's a couple of rings of microtubules called the polar rings. Within that, you've got the conoid made of these spirally arranged microtubules and microfibers. And then radiating from that, you have microtubules that run along the body axis and are probably involved in locomotion. But there, those rings of microtubules are what make up the apical complex. And again, that's important in attaching to host cells. You can see some of the complexity of it there. That's a negatively stained transmission electron micrograph. And you can see those polar rings, and then that kind of crosshatched plaid-looking area is that spiraling ring of microtubules. And beyond that, you can see the subfolliculars. Right, anyway. This is a complete cross-section through toxoplasma inside a host cell, I might add. The apical complex is on the right, and that conoid is labeled C. Attached to the conoid internally, you have little filaments called micronemes, and then elongated sacs. They're labeled R in that picture, called rhopteres. And the micronemes and the rhopteres secrete proteins that allow the parasite to attach and penetrate. And then the nucleus is kind of in the back half of the cell. There's a Golgi on top of the nucleus, between the nucleus and the rhopteres. Right. Lot of annoying detail, but again, understanding how these things are put together is one of the clues to trying to beat them. And I think I mentioned that malaria currently kills an estimated something like 450,000 people each year. Used to be even higher than that. So yeah, this is globally one of the killers. This is one of the things that Bill Gates is spending an awful lot of money on trying to stop. It's not that big a problem here. Arkansas, I think, sees fewer than 10 cases a year. But in many parts of the world, it is an absolute scourge. And we'll get to that in a bit. Right now, there's that apicoplast. And I mentioned that dinoflagellates evolved at this weird thing where they'd engulf algae and dismember them and steal their chloroplasts. Well, there are some cases where a protist engulfed an alga and didn't dismember it and kept the chloroplast anyway. So this apicoplast right there is surrounded not by 2 membranes, but by 4. And that tells you that the apicoplast used to be a complete eukaryotic alga. The outermost comes from the ancestor that engulfed it. And then the second outermost is the original plasma membrane of the symbiont, of the eukaryote. And then the inner 2 are the ones that belonged to the chloroplast. So you've got a symbiont in a symbiont in a symbiont there. I don't know if I'm explaining it that clearly, but yeah, there's this weird evolutionary history of apicoplasts. And again, they still contain their own genomes. What they do, I'm not sure, although the protists can't live without them. With a little bit of luck, you might actually see these guys tomorrow. The easiest group to study of apicomplexans is a group called gregarines, which do not parasitize vertebrates. You can't get infected by a gregarine. They only infect invertebrates, mostly insects, although some will infect others. And they're what we call monoxenous. Xenos is Greek for a guest. So if you're afraid of having guests, you're xenophobic. Monoxenous means one guest. Actually, it also means host. They use the same root for both. And monoxenous means that they only have one host in the life cycle. The entire life cycle, or almost the entire life cycle, is spent only in one species. Very typically, you have this cell that's got a septum that divides it into 2 major parts. So you can see these cells look like they've got kind of a dome-shaped anterior head and then a body stretching along behind them. That's typical of gregarines. And then up at the front end, there's this funny-looking thing that's got little hooks and fins and things. That normally is buried inside the host cell, and it's an attachment structure. These things will stick themselves onto the outside of a gut cell in an insect or other, whatever the host invertebrate is, and use that penetration structure to hook in. If you must know, that anchor structure is called an epimerite, and then back of that front half of the cell itself is the protomerite, and behind it is the deutemerite. And again, not of much ecological importance or economic importance. Many of them don't seem to do their host very much harm, but they may be useful as model systems for understanding other apicomplexans a little bit better. They'll certainly be the ones that we have the best chance of seeing tomorrow. Coccidians have a new wrinkle. They can infect vertebrates, and they are generally intestinal parasites like gregarines. Toxoplasma is one that can infect humans, although that's usually asymptomatic. Where we encounter them most is as parasites of dogs and cats. I've had students that were vet techs before, and apparently coccidiosis infection is fairly common in puppies. It's one of several things that can give puppies diarrhea. So if your puppy has diarrhea, please take them to the vet. Cats can get coccidiosis as well, and it's a particular problem in poultry farming. That particular one that you see right there are cysts of the coccidian Emeria that was isolated from chicken poo. And again, this is the kind of thing that can wipe out a pretty good chunk of your flock if you're not careful. This is important because the Tyson chicken is, after all, the state bird of Arkansas. Right. Okay, if you eat one of those oocysts, they produce a generation of cells called the sporozoites, which penetrate host cells and repeatedly divide. That's what you're seeing, what the arrows are pointing to. You're looking at a host cell that's been penetrated by a sporozoite, and the sporozoite has repeatedly divided. So you have this mass of daughter cells called a schizont. And when the schizont matures, the host cell ruptures, and the little swarmer cells come bursting out, and they go on to infect more cells. So they'll go through several rounds of asexual division inside their hosts. And the problem is this can often happen in sync. So imagine you're a chicken, and a whole bunch of your intestinal cells all spontaneously rupture at the same time, releasing lots of schizonts, and then the same thing happens again in 4 days. You get these relapses of symptoms that can be very severe, and in chickens are often fatal. There's several rounds of this, and then the cells go through a sexual stage, forming new oocysts like this, and round goes the great circle of life, that alternation between asexual and sexual reproduction. Oh, sorry, that's kind of gross, but that is digestive ceca of an infected chicken up at the top and an uninfected control down at the bottom. You can see the lining of those pouches is red and swollen and has blood clots on it from hemorrhages. So yeah, this is not something you want your chickens to get. And then some coccidians actually alternate between multiple hosts. Toxoplasma gondii alternates between cats and rodents. It forms cysts in the cat gut. The cat sheds them in feces. If the cat feces or something contaminated with them gets scavenged by rodents or sometimes birds, the infected rodent or bird gets these cysts in its muscle tissue. And then when the cat eats the rodent or eats the bird, the cat gets infected, and round goes the circle of life. Humans can get these in as well in the muscle tissue and sometimes even in the brain. About 50% of the human population is infected with Toxoplasma gondii. In most cases, it has very few symptoms, but it can be bad if you're immunocompromised. It can also cause birth defects because it can cross from a mother to a fetus. So this is the reason why my wife immediately stopped cleaning the litter box and made me do it as soon as our son was born. Now that he's 9, I should probably ask if she wants to renegotiate that. But anyway, there's another group called pyroplasms, and these guys are blood parasites instead of gut parasites. They're spread by blood-sucking arthropods. In the middle of this slide and a little bit to the right, you have the thing inside one of these blood cells that looks like a cross. Those are that's a pair of little swarmer cells that will burst out of, not a pair, it's 4 little swarmer cells that will burst out of the red blood cells and infect more. This is still a problem in many parts of the world because it causes a cattle disease called red water fever, which has been eradicated from the United States, but it took a long time and it still could come back. Some earlier stages of Babesia right there are visible in other cells. Cells that have just been parasitized have a stage inside them that looks a little bit like a diamond ring. So pretty close to the dead center of that picture, you can see a red blood cell with a ring stage. And then when they undergo this merogeny process is when you get 2 or 4 pear-shaped cells, which is what I talked about at the beginning. Right. And then finally, malarias are caused by a group called Hemosporidia. And like pyroplasms, they alternate between invertebrate and vertebrate hosts. An infective stage called the trophozoite is injected by infected insects. In the case of human malaria, it's a mosquito. And the trophozoites penetrate into blood cells. Over on the left, you've got an early stage of the infection. The cells are the parasites are pretty small. And at this stage, they've got a nucleus that stains strongly and a central vacuole that doesn't stain. And the upshot is that they look a little bit like diamond rings inside the blood cells. This is actually called the ring stage of malaria infection. They'll go through the ring stage and then they'll start dividing asexually, a process called merigony. There you've got on the right a blood cell that's just about to burst because it's packed full of these asexual swarmer cells they're called schizonts. Don't worry about the name. And those will come bursting out of blood cells and also of liver and spleen cells. And they have this nasty habit of doing it in synchrony. So if you're infected by malaria, what happens is that a whole bunch of red blood cells explode at once. And these swarmer cells called merozoites go bursting out all at the same time. And that dumps byproducts into the blood, the rubble of destroyed red blood cells. That in turn triggers an immune response. So once every 3 days or once every 4, depending on what species you're infected with, you get hit with a few hours of chills and then hours of high fever and the headache from hell, and then a phase of sweating until you finally worked your way through it. It's not pleasant. It can cause chronic anemia, and this is particularly bad in kids. If you have, anemia, that means you don't have enough blood cells to carry oxygen, not just to the body, but especially to the developing brain. So childhood anemia can leave you with some cognitive deficits. And of course, this is a big drain, not just on personal well-being, because it really sucks to have chills and fever and headaches. It's also a drain on economic productivity, because if you have this, once every 4 days, you can't work, you can't go to school, you can't really do anything but wait for the symptoms to pass. In every round of this, a few merozoites, those swarming cells, most of those will just penetrate new blood cells and repeat that asexual cycle over and over. But in every round, a few merozoites will transform into gametocytes, which look like this. They look kind of like bananas. And that's the stage that infects a mosquito that feeds on your blood. If a mosquito bites an infected person, it will slurp up some gametocytes, and those undergo a sexual stage within the mosquito. I'm not going to focus on the details of the sexual stage, but that sexual stage yields a new generation of infective cells that pass to a new human host when the mosquito bites somebody. So the sex all takes place inside the mosquito, and asexual reproduction takes place in an infected human. By the way, there's certainly a lot of research into vaccines, although that's turning out to be a very difficult task, and into anti-malarial drugs. Probably the simplest way to cut down on the incidence of malaria is basically to use lots of mosquito netting. And there are actually some charities. If you have any money left over after donating to, coronavirus victims and protester bail funds, there are some charities that provide cheap, locally made, but very effective if used properly, insecticide-soaked mosquito nets to parts of Africa, for example, where malaria is a severe problem. And as long as the nets are being used right, they're effective. They stop malaria transmission just because they break the chain of transmission. And one of the things I talk about in my parasitology class a lot is if you've got a parasite that you need to control, it's often cheaper and more effective if instead of finding a drug to poison it with, you come up with a way to cut off its life cycle, keep it from completing that cycle, and you can, in at least one case, drive it nearly to extinction. So yeah, nets for people is one way of bringing down that death toll, and people are actively working on supporting that. I said it kills about 800,000 people annually. I think that's an outdated slide. I'll need to fix that. It's kind of hard to estimate because malaria tends to hit in tropical countries, Africa, South America, and tropical Asia, which are also in some cases underdeveloped countries that don't do a very good job of keeping statistics for various reasons. So last I looked, the number of deaths per year had gone down from about 800,000 to something closer to 450,000, but there's still some uncertainty about it. Malaria actually killed 60,000 US soldiers in World War II. Soldiers were issued anti-malarial tablets and mosquito nets and things like that, but it was still a real danger for them. So there you've got a poster warning soldiers against going to bed with a malaria mosquito. I'll just finish with this because it allows me to recite some poetry. Sir Ronald Ross won the Nobel Prize for Medicine in 1902 because he worked out that complete life cycle under very difficult conditions while serving in India when India was a British colony with the Indian Medical Service. It's fairly straightforward to study malaria in infected humans. All you have to do is have some patients, some infected people who are willing to let you draw their blood. Finding out what it does in the mosquito is a lot more difficult. Ross originally thought malaria was caused by intestinal poisoning, but he studied in England with Dr. Patrick Manson, who hypothesized that it was transmitted by mosquitoes. Great hypothesis, but actually testing it is difficult because you have to learn how to dissect mosquitoes. And Ross was working in India in the summer, and parts of India, if you're not in the mountains, India is pretty damn miserably hot in the summer. Working in a room with no air conditioning where he had to turn off the ceiling fan because, you know, when you're dissecting a mosquito, the slightest air current can blow away your entire work. So he spent a very long time extremely sweaty and thirsty and uncomfortable. And on the day he confirmed that malaria was transmitted through mosquitoes by finding that sexual stage inside mosquitoes, he actually wrote a very nice poem. This day relenting God hath placed within my hand a wondrous thing, and God be praised. At his command, seeking his secret deeds with tears and toiling breath, I find thy cunning seeds, O million murdering death. I know this little thing a myriad men will save. O death, where is thy sting? Thy victory, O grave. So, yeah, Ross was actually kind of a jerk and used political pull to keep from having to share the Nobel Prize with an Italian researcher who'd found much the same thing and in some ways done a better job. But yeah, he did write poetry about his discoveries, and that's rather a nice example that I can show you. Okay, so there, that would be that. Let's see if we have any questions on the... Oh, Savannah says many of her chickens died from coccidiosis, I assume. Okay, well, I'm terribly sorry to hear that. But yeah, it's a problem. Do you have like really is just just some yard birds, or do you have a you have one of those big Tyson operations, or just a small flock for eggs every morning? While I'm waiting for an answer, Wendy says I can buy a dinoflagellate dinosaur that I feed and shake up, and it lights up when I shake it. Okay, cool. I'll maybe, if I ever have money that I feel like spending on that, maybe I'll look into that. Dinoflagellate dinosaurs. Now, I saw a cartoon that I hesitate to mention, but a company in, of course, Eureka Springs used to sell nerdy t-shirts, and one of them was a picture of dinoflagellates, and it was dinosaurs dressed in black leather holding whips. I don't get it, but it seemed funny. And Savannah says she's got just a few silkies. Okay, well, I'm sorry you lost your silkies, but yeah, this is a problem. Okay. I'm going to go ahead and stop recording now. So wait for Hello Cat.